Welcome, everyone. Thank you for joining us today, especially at the end of August as the summer closes and the new year begins. Marie, we'll let you start.
Perfect. So thank you guys for all joining. So I'm Maria, I'm going to be moderating today's session. So just wanted to quickly make a quick introduction to our guest speakers today. So next slide. So leading the session will be Dr. Olga Kubassova and Dr. Anitha Singareddy. Starting with Olga, she is the CEO and president of Image Analysis Group. A little bit about her background. She's a mathematician and computer scientist who founded IAG in 2007, following her PhD at the University of Leeds in the UK. So she's been leading this for 20 years, pioneering mostly AI-driven medical image analysis, using the technology applications DYNAMIKA and novel endpoints in trials. So far, with 120 plus publications and multiple Entrepreneur of the Year awards, Dr. Kubassova is the CEO and president of IAG. Then leading over to Dr. Anitha. She is the head of scientific and medical affairs also here at IAG, a board certified physician with 15 plus years experience in clinical research and oncology imaging. She's an expert at using standardized and advanced imaging modalities and associated endpoints in clinical trials, advising some of our leading biotech and pharma clients across medical and scientific group of experts. So just a little bit of housekeeping. So this session today will be about 45 minutes, and we'll also open to Q&A at the end. However, throughout the session, feel free to just publish any questions you might have, and we'll come to them all at the end accordingly. This session will also be recorded, and we'll be sending the recording out after to all of those who attended, but it will also be available on IAG's official YouTube channel, which you can see here. Do advise to subscribe as we constantly update new publications on there, so it keeps you up to date with anything new and fresh in the clinical trial space. Should you be interested in any of the things discussed as well, we'll also be offering one-to-one DYNAMIKA demos at the end of the session, and also just in-depth expert discussions with anyone in our team. So we have our contact details here as well, contact@iag.group.com. But yeah, so thank you so much as well for all the registrants as well. I know there's a lot of demand for this session, so look forward to diving in. So what to expect today, we'll just do a warm introduction and welcome, discuss a little bit more about IAG, and then go into the deep depths of designing imaging strategies and using AI and advanced endpoints as well that support clinical trial enrichment and investor confidence. We'll then talk through an operational playbook and then close on a Q&A. Thank you.
Oh, thank you, Marie. And welcome everyone again. So we had quite a lot of people registered for the webinar, so I expect people will be coming in. So as we continue, I will start by introducing Image Analysis Group to those maybe who are new to us. We are specialist imaging CRO. With just about 20 years of experience supporting biotech and pharma clinical trials that use central imaging with and without AI-enabled workflows to assess safety and efficacy of their drug candidates. The company started with a foundation in image processing, so we were building proprietary algorithms for trial-based image analysis, and hence the image analysis as a name of the company. We then developed a DYNAMIKA platform as a secure infrastructure to bring together all of those image processing algorithms, but at the same time enable reliable data collection and reliable workflows to actually process that data for the purposes of AI, for the purposes of trials, and so on. So today we provide end-to-end services, technology, and expertise to sponsors who will be using medical imaging across their clinical trials. Our operations are across UK, where our headquarters are, Europe, US, India, and also Australia, China, and Japan, where we have partners. IAG team normally works alongside the CRO or sponsor team and supports everything from protocol development, imaging site selection, qualification of those sites, assessment of the data, so quality control of the data, and then centralized reads. We work in multiple therapeutic areas, and today we will be giving you quite a few examples from those. But our core expertise lies in rheumatic and autoimmune diseases, so autoimmunity in general, musculoskeletal conditions, bone health, oncology, neuro-oncology, neurology, obesity, and cardiometabolic health. Over the years, we built a global network of sites and radiology readers, as well as partners who contribute to the trial success which IAG runs. So what it means for our sponsors, it's a direct access to experts, it's very fast study startup, it's very reliable central review, and of course, a scalable imaging operations, which needed for many of the programs. I wanted to also emphasize that, today, what imaging CRO is really changing, and being a modern imaging CRO has its own requirements. What it used to be is call up or maybe a team of radiologists who would be analyzing data by viewing those images, often deploying third-party platforms. Today, it's paramount to have your own technology platform, which is auditable, which is end-to-end solution for any kind of trials. Of course, the team itself is also important, and access to that data is also very important. Instead of maybe a siloed radiological reviews, which are disconnected and disjointed a little bit, you need to have access to the data from any kind of angle, from imaging scientists, biomarker scientists, software engineers. So you really need to have a team model which is working alongside the platform which supports your technological innovation. Then, of course, comes AI capabilities, and maybe this is relatively new, but it's certainly becoming extremely, extremely important. So if AI workflow is just bolted on top of a legacy system, and AI is kind of just thrown as a marketing word, I don't think it's going to deliver much value. When you use AI as part of the trial, and you're delivering your results with additional AI elements, which can simplify or supplement some of the human steps, it makes a world of difference. And the same for trial lifecycle roles. So when do you get engaged to deploy AI or deploy any advanced imaging efficiently? Most likely, to make the most impact, you really need to be working on a protocol, implementing the protocol endpoints, assessing how the sites will be able to acquire images which lend themselves for AI processing. You can't just be pulled sometime after protocol is locked and everything is decided. And of course, what it means for all of us, and especially for those who are developing drugs, is that you have a single point of access to your data. You have a team which is accessing the data, and it's not just IAG team or site team, it includes sponsor team and partner teams. You now have a centralized data hub surrounded by services. And of course, that makes a world of difference if you want to do anything advanced in a trial. So we provide end-to-end services within the imaging clinical trial. So as I said, the first and foremost is really clinical protocol review and development of the imaging strategy, which will then support how the endpoints are being read and implemented and how they're presented to the regulators. Then looking at the sites, feasibility of the sites, training of the sites, making sure they acquire the data in the right format. Then, of course, collection of that data, quality control of that data, and then amplification of the data with AI at some of the steps, which allows us to do efficient central reviews or efficient eligibility reads at the time of recruitment of the patients. I will move to just give you a schematic of our DYNAMIKA platform, because we will mention it quite a lot. DYNAMIKA is something we created in-house. It's an enterprise level platform, which basically can do many, many things and manage many, many stakeholders. As a data source, we can take any kind of data source. As you know, the trials nowadays are incredibly complex. So an obesity trial may incorporate a DEXA scan for bone health, a liver scan for liver fat, an thigh MRI, and ECG for cardiac safety. The same for oncology trials, anything from photographs to MRI to PET scans. So the data itself is getting richer and more complex. It's also flowing from different divisions of the hospital. So some of it is acquired by radiology department, some of it acquired by PIs, some of it acquired by cardiologists. So you could see that data sources being diverse and data itself is being diverse. So the very first thing is to ensure that the platform can take any kind of data. It's anonymized, it's safe, it's secure. It could flow into the platform worldwide. So our sites could be located anywhere from US to China, and that wouldn't matter when the data flowing in. There are operational safeguards and guardrails when you're going into data-sensitive geographies. Once the data is in, you have kind of an engine which takes care of the data, from quality control, query resolution, central review, data import, export, and how you're interacting with different partners, how to deploy the partners to process the data. And of course, then how do you control the data flows? So you have an operational, we call it operational tower of control. So you have reports, you have audit trails, you have data exports, metrics, and so on. And all of that, the enterprise level of the system, is deployed on a trial-by-trial basis. Every single trial will have a multitude of stakeholders. Just the sites alone would be between 10 and hundreds of sites, depending on the size of the trial. The sites could have a different equipment, but everything related to the site has a specific workflow. For instance, if the sites uploading the data, they can only see their own data in a very secure way. They also get data anonymized. They also have ability to interact with our team internally about the quality of the data. On the other hand, an imaging specialist who is reviewing data from all of the sites and assigned to the study will see data in a comprehensive way. So they will see it study by study, or they will see it modality by modality, the way the study is set up. But their workflow is completely different. If we come to the readers, here you can have access to the data as a reader, where you will be making decisions using either your judgment or AI tools, or a combination of your judgment and AI tools. And again, you can have several readers accessing data simultaneously, reading it simultaneously, and adjudication happening on the background. So again, it's a very different workflow. It's a very different setup. Obviously, nothing to do with the site workflow or imaging specialist workflow. And lastly, you can have sponsors themselves who also need to make decisions based on the data. So various dashboards, various information and kind of KPIs around site performance or data quality. All of that becomes available not just on a monthly basis, but in real time. So the enterprise level nature of the platform and ability to bring various stakeholders into the trial under a single umbrella gives you ability to really have an operating system above your trial, like we're operating our computers. Now, of course, if we're talking trial on trial, we can design a program. So our DYNAMIKA platform works across programs as well as trials. So we can separate all of the trials under one sponsor from another sponsor. We can separate all of the trial in a particular indication. And this allows you to build a very reliable data pool and then take it one step further with AI, which is coming up. We see AI being kind of one of the stakeholders in the trial, really. So AI could come in many shapes and forms, as we all know, but why not allow AI processing to sit as part of our procedures? So we optimize the way we deploy various tools and various decision-making tools in the clinical trial workflow. And AI could be used for anything from patient stratification to building better workflow for the readers, to overarching analysis of the data post-trial to depict certain manifestation or impact of the drug on patient subgroups. There are different types of AIs, as you already probably know, and I like to separate them in these three. One is probably the simplest, is automation. And this really relates to the process automation or efficiencies we can gain. And this is very simple. You know, if we take Viva Vault or Medidata, this is kind of automatic document routing or visit tracking. With DYNAMIKA, you have the same thing, but for the imaging. You automate certain steps, certain notifications, you flag events, you distribute data correctly, you make sure that that trial goes this way, this is the assigned team. Even the way notifications are done for different type of events or different reports flow into a different kind of stakeholders, that would be all process automation and efficiencies. Second type of AI relates to data processing, data analysis. So normally it would be a very specific narrow task. For instance, segmentation of organs in a CT scan or MRI scan, or identification of a certain threshold or barrier. So we have a library of AI tools which are specific to a particular indication. Of course, for oncology versus scleroderma, you would have a very different need because you're looking at very different scans, you're looking for different images, different changes. So you have a different tools which allow you to segment or align or quantify. And effectively, it reduces burden on radiologists. So as you reduce burden on the radiologist, you minimize the time needed on the image, which directly translates in a commercial benefit to the sponsor, which aligns very well with our all overall incentives because we're getting more and more images every day. We also have a number of great partnerships with AI companies who develop technologies which allow us to do things faster or better, and we API them into DYNAMIKA as part of our AI pre-processing of the data. In this case, AI will be deployed to pre-process the data, and data with AI results will be displayed in front of the radiologist who will make a final decision and sign off the report. Last, and probably the most interesting type of AI, which we are all fascinated about, is a general AI or generative AI. And here is where you can combine data from a single trial, not necessarily just imaging, or you can combine data from multiple trials. So, of course, you know about building synthetic arms for the trials. You're looking at subgroups. You could even look at overarching data from a large phase 3 program to predict certain events. So when you start looking at the multitude of data and you're predicting an event, that is generative AI, which allows us to make a decision. What is the likelihood of that decision based on the available data? We will show you a couple of examples based on that. And again, we have a number of partnerships with DYNAMIKA being a central platform and AI routines being available as needed on specific trials. So what type of AI and what tool to select? I think in trial, it really depends what question needs to be answered. We're going to go phase by phase, showing you the AI in practice and show you the real examples of how we're deploying AI in our group. So in phase 1, generally, we're looking at more interesting way of selecting dosing or building a targeted compound profile. So that's where AI and biomarkers really driving those decisions. In phase 1B or maybe 2, this is where we build in a differentiation of novel drug candidates from one another, or we build in a more comprehensive understanding of a mechanism of action. So very different type of AIs actually being used here. And again, when we come into phase 2B or 3, now we're going into global logistics, which of course complicates any kind of analysis, which we're bringing into the AI. Medical imaging here is a very important point because it really helps us to de-risk the trials. It's another data point which addresses something very specific, very structural, and allows us to be very precise with our decisions. So in clinical trials alone, with the comprehensive data pool could be split into what question needs to be answered, and AI tools can be applied to it. I pass to Anita.
Thanks, Olga. Many phase one clinical trials already include imaging to monitor the disease and also look for the early signs of treatment efficacy. So when we look at phase one, the focus is primarily on, the safety and tolerability, the dose selection, and early evidence of treatment activity. So the challenge here is that these decisions often need to be made with relatively small patient cohorts, and also while the study is still ongoing. So this is where AI and quantitative imaging can add another layer of information to what actually the biotech or the pharma companies are looking into. So rather than relying only on the conventional 2D measurements, we can extract more detailed information from the routinely acquired images on a trial that could include the volumetric changes, the changes in the tissue characteristics that may not be captured by, the standard response criteria alone. So this can provide earlier insights into the treatment activity, the biological effect, and also the potential toxicity. A good example is that, as a company, as IAG, we have supported a phase 1/2 GBM program where AI-enabled automation and quantitative imaging analysis. It allowed the imaging data to be processed much more efficiently. So this meant that the interim data set was available earlier, helping to bring the interim decision towards around 3 months. So this is more about extracting the more information from the imaging that has already been collected and used to support the faster and more informed decisions on early clinical development. And that is why we feel that AI plays a pivotal role in the early phase clinical development program. And moving on to the next slide. So as you can see here, we have announced a number of partnerships. And what I think is interesting is here is they represent quite different applications of AI and quantitative imaging across drug development. So the indications are very different on these trials, but the underlying question is quite similar. Can we get more useful information from medical imaging? So with Genenta GBM phase 1/2 study, so the challenge here is around the response assessment. So as we all know in GBM, an apparent change on MRI does not always tell us whether we are seeing the true progression or the treatment-related effects. So in this case, the conventional assessment is combined with the quantitative and AI-driven tumor analysis. So that can provide a clearer picture of what is happening over the time. And then again, in the Avalon study, the AI-based analysis was used to quantify the imaging changes and look at the differences in the treatment effect between the dose groups. And DAVIX, this takes a step further, and it is an example of taking an imaging biomarker from the scientific development and then translating it into something that can be standardized, it can be reproduced across sites, and ultimately it is used now in a clinical trial setting. So again, to make this a little more tangible, let's take GBM as an example. So GBM is particularly interesting because what we see on a routine MRI scan is not always straightforward to interpret. So an apparent change may represent a true tumor progression, but it could also reflect the treatment-related changes, including the pseudoprogression. And you could see that the complexity, which is being illustrated on this slide. So what may initially appear as one area of abnormality can actually contain several different components. So it can be the enhancing tumor itself, the surrounding edema or the flare abnormality, areas of necrosis, and other treatment-related changes. So the idea here is to move beyond looking at it as a single abnormality and then start categorizing these different components separately. So alongside, with the established frameworks like RANO and now RANO 2.0, we can use advanced imaging and quantitative analysis to look at the tumor volume, the flare changes, edema, and potentially the other imaging characteristics such as perfusion and diffusion as well. So particularly in early phase GBM studies where patient numbers are small, extracting as much as meaningful information as possible from every scan, it can be extremely valuable. So moving on to the next slide. So yeah, as we walk through the phase one and the importance of AI in the phase one clinical trials, so the key point here is that AI can help shift the phase one imaging from simply monitoring the outcomes to actively support the faster and more informed decisions. And also the value doesn't necessarily stop at phase one. So the insights we generate early in the development can then help inform how we approach the next stage of the program and the later phase trials. So as we move into the phase two, the opportunity is to use the data to design the imaging strategy around the key development and also the questions that we're trying to answer, rather than just relying on the modalities or the assessment methods that have been traditionally used. So in other words, we can start with the questions we need to answer and then build the imaging strategy around that in the early phase trials so that you can have the informed decisions made in your late phase trials.
Thank you, Anita, very much. I'll pick up and just give you another example, which is now speaks about the case study, a work we have done with Eli Lilly a few years back, and the reference here in the corner. And what was interesting here is that one of those autoimmune conditions, rheumatic disease, rheumatoid arthritis, where you have an opportunity to acquire a very solid imaging time point. So what I mean by that is patients generally getting contrast agent, which enhances tissues which are more perfused, like synovitis or edema. And here in a screenshot, in the screen, you see the pre-contrast and then post-contrast images with synovium is being whiter and non-inflamed tissue showing in the dark colors. This is a very complex acquisition needed to separate patient subgroups or really understand the dose of the treatment. And this is where we found ourselves, where you take in images like that. What do you do with them? One of the options is just to look at that data. Radiologist or an expert can look at this data and using a scoring system, which is a radiological expertise-driven scoring system, would say on a scale from 0 to 3, do I think this joint is affected as 0, healthy, or 3, which defined as the worst imaginable joint. And of course, you can imagine that the worst imaginable could mean anything generally to anyone. And of course, there is a specific training associated with the scoring system. But what if acquisition wasn't done correctly? So that was one of the problems which we first saw in a cohort, because that was a big retrospective cohort which came to IAG after there was a challenge to separate doses of the vorasidenib, so anti-inflammatory treatment by Lilly. What we realized that an image acquired at some point and the same anatomy acquired 9 minutes later could look quite identical. And the only time you could see that the acquisition was done in the wrong time frame is only DICOM header. But we had about over 400 time points. So looking by hand at the DICOM headers is not going to help. So running an AI algorithm, running automation, we were able to distill that actually about 10% of images in a metacarpophalangeal joint and about 4% of images were acquired completely incorrectly. So retrospectively, we had to exclude those data from the common cohort and actually define a better protocol, so standardize the protocol for rheumatoid arthritis acquisition. Second point, instead of looking and saying 0, 1, 2, 3, we needed to develop a much stronger quantitative scoring system, because if you look at the numbers, placebo 32 patients, baricitinib 1 milligram 21 patients, 25 patients for the next dose. So very small cohorts. How do you achieve statistical significance here? And the only way to look at the data quantitatively is to deploy a system which allowed us on a pixel by pixel basis, analyze those images. And you could see here what a difference you could see from baseline to week 12 to week 24, where the inflammation completely goes. It's an example of one of those scans from the trial published in this paper with a reference below. Once you have quantitative data, quantitative method applied to correctly acquired data, it's really irrelevant how many images you have. You will see the change. You will definitely see it visually, and in this case, statistically significant. And of course, what's remarkable about it, of course, that data was going straight to the regulatory authorities. And that's an evidence which is very, very important.
So we've talked about, the potential of AI and some of the ways in which it can support the clinical development. But I think it's also important to step back and look at why there is such a need for better tools and better approaches, particularly in the early phase trials. And if you look at the overall probability of success in the drug development, the phase 2 really stands out, as you can see on the slide. So around only 29% of the program successfully moved from phase 2 to phase 3. So making this one of the biggest hurdles in the development pathway. And this is particularly challenging because phase 2 is where we're often trying to answer some of the most important questions. Are we seeing a real treatment effect? And which patients are most likely to benefit? And are we using the right endpoints to detect that effect? This is where AI can start to have a very practical impact. So, for example, AI in advanced analytics can help us better characterize and stratify the patients, potentially, identifying the subgroups that are most likely to respond, and in some cases, helping rescue a signal that might otherwise be missed in a heterogeneous patient population. So it can also support more efficient and consistent assessment of endpoints. So in imaging, for example, an automated or AI-assisted analysis can reduce the variability, and it can also extract more quantitative information from the data. Next slide, please. So taking this idea one step further, so this is where AI can really change how we look at the medical images. So one practical example is our partnership with Picture Health, which is focused on making AI-enabled imaging biomarkers more accessible and practical for the use in clinical trials. And more importantly, this is based on the routine imaging already being collected in. So depending on the cancer type, as we all know, the routine imaging may be a CT scan or an MRI scan. So from these images, we can derive a range of quantitative measurements and additional insights. So starting at the top left, one example is the volumetric tumor burden. So with the conventional response assessment criteria, such as RECIST and iRECIST, we typically assess tumor burden based on, the longest diameter of the lesion and some of those measurements. But using AI, we can take this a step further by assessing the overall tumor burden volumetrically, rather than just focusing on the selected target lesions. So we can also assess the tissue surrounding the tumor. It's more of the peritumoral environment, providing the additional information about the changes occurring within the tumor microenvironment, and also including those related to the treatment and the immune cell infiltration. Another feature is the tumor margin sharpness, which helps characterize the tumor boundary, and it may provide insights into its potential behavior and its spread. And also, we can look at the tumor heterogeneity, as greater variation within a tumor can be associated with a more aggressive disease. And then we can also assess the vascularity, or the characteristics of the blood vessels around the tumor. Since the tumor growth is associated with angiogenesis, the complexity of the vascular network, it can provide additional biological insights and also has been linked to the human resistance. So essentially, we're taking a routine imaging that we already acquired in a clinical trial, which is already a part of the study, and we're looking beyond the conventional measurements to extract that additional information about the tumor and its biology. And then moving on to the next slide. So this is one example of what we mean when we say we can look beyond the image itself. So if you haven't heard about it already, so this is one of the Pictures Health standard imaging biomarkers, which is called the QVT score. It is the quantitative vascular tumor score, which is a biomarker for tumor vascularity. So we start with a standard CT scan, and from that, we can compute more than 900 different metrics. So these metrics cover several different aspects of the tumor vasculature. So rather than simply measuring the volume of the vessels, feeding the tumor, we can also assess the characteristics of the individual vessels, such as the volume, the curvature, and the torsion as well. So as you can see on the left on the slide, the patients with the low QVT score, they tend to have vessels that are more open and less tortuous. And these patients generally show a better prognosis across, the range of treatments. Again, patients with a high QVT score shown on the right, they tend to have vessels that are tighter and more twisted around the tumor and generally have a poorer prognosis. And the other advantage is that we can also assess these radiomic features longitudinally over the course of treatment. It's not just for one time point scan. So, for example, if a patient starts with a high QVT score and the vasculature doesn't begin to open up during the treatment, meaning that the score does not shift from high to low, that may be an early indication that the treatment is not working. Similarly, if a patient's QVT score changes from low to high over the time, this may also suggest that the treatment is no longer having the desired effect. So potentially, we can see the changes even before we see more obvious changes in the tumor size, and that is where such advanced image analysis plays a pivotal role in having those informed decisions on your early phase trials.
Thank you, Anita, very much. And I think these very specific biomarkers and very specific case examples, they could really be extracted from very early phase trials, but also the trials where you just look for efficacy. It could be a late phase trial with a subgroup, or could be a real world evidence study. And I want to bring another example here where we were developing as a partnership with Takeda. Takeda is a Japanese pharmaceutical company, an AI-driven index to recognize the impact of their drug, which was already on the market called Entyvio, on patients with inflammatory bowel disease. So what we were doing, and I put a reference here again, so you could see that that article was the top-cited article of 2025, which of course tells us about the level of interest bringing this type of approaches into clinical trials. Here in this partnership, what we were able to do is to look at the histology slide, which is taken as part of the routine colonoscopy, and assess that histology slide using AI tools developed in-house. So first of all, every slide looks slightly differently because the biopsies are taken at random places. And you could see that here in this case, we are splitting the slide into separate tiles. So each tile then processed using AI to understand the different cell types, their geography, the combination of cells. And then as soon as the tiles are identified, we can run the AI methodology per tile. So now you have the whole histopathological slide from a patient, and we're looking at the cell types. Once we identify that, it's all happening on the same infrastructure as we're using for phase 1 or phase 2 or phase 3 trials, meaning that you can run your trial and then retrospectively really look back at those trials and understand how you can deploy the same data with more techniques, more methodologies. So the DYNAMIKA software, our platform, was able to connect sites from around the world who were sending histology images and the AI models which were running on the images to really identify the cells. Why we found that fascinating? For the very first time, we were not restricted by the size or location of histopathological slide. We could just take a sample and process every single piece of that sample. A pathologist would normally have a high resolution view and focus on a small piece of the slide. Here, AI really doesn't care. So you could see here an example of a biopsy histology slide, digital slide, processed using AI with indication where potential problems are. So 0 being green and red being non-C4, which is the most effective, the most severe place where there's potentially ulceration. We can further distill it into numbers. So we could say how many tiles have what kind of score, how many cells are per tiles. And then we can derive a single score, which defines the patient individual signature. And I want you to notice here that the signature here is 4, meaning the patient is severely affected. When we processed another biopsy, which also gave us a score 4, but the distribution of the disease was completely different. The reason the score 4 is on both patients is because pathologists would be looking, if there is even a single ulceration area, it would be scored 4, so the worst. But the difference, the starting point of the slide, as you could see, completely different. You can also see how many of them were non-C, 0, 1, 2, 3, and 4 within a single biopsy. So of course, what does it mean for the drug developers? It means that you could really look at the change from screening to a follow-up visit, now not on a very general 0, 1, 2, 3, 4 scale, but you can go down to a cell counts where you could say that a delta change in the number of neutrophils per whole slide biopsy is such. And of course, that gives an unprecedented level of precision. And this could be achieved not just with histology, but with any other type of imaging. Once you deploy thinking around, what do you actually want to understand? Do you want to understand how fast your treatment works? Do you want to understand the earliest possible changes? Do you want to see cohorts? And I think it's a great example that any type of imaging with the right tools, the right thinking could play a significant role. I will pause on all of the examples, and in the next couple of minutes, I'll just give a very quick overview of what we think operational playbook may be, because we notice that there are a lot of chief medical officers and operational executives joining this webinar. So I think here, the primary challenge of AI doesn't lie in the fact that there are tools. It really lies in integration of AI into clinical trial process. And I think what could be done, what could be thought about to actually make use of AI? Obviously, the right resources and the right partners, the right digital infrastructure to prevent any data leakage or data loss. Absolutely data availability. So you need to have available data and regulatory framework around this data. But I think what's the most important is a digital mindset. And I think from about maybe 12, 18 months ago, when ChatGPT just appeared and made such a boom on the market, I think it's quite important to realize that our mindset is moving towards digital in clinical trials as well as everywhere else. So if we implement the right tools and start slowly but surely thinking of technical, quantitative use of advanced technology as we're planning the trial, I think it will help as we're progressing along. We also need to think about how to collect the data responsibly, and of course, how to standardize that data. DICOM is a very complicated standard. And of course, every scanner will generate slightly different images, which need to be collected uniformly. You could think about not just a single trial data, but a multi-trial data, which of course makes the guidelines for each of the trials, they need to be harmonized and unified. Data privacy and security is, of course, a huge thing, and there are a number of guidelines now and procedures which allow us to really think about image anonymization, patient confidentiality, way before any trial starts. And today, we're using our DYNAMIKA platform under various standards and maintain those standards to make sure that the data gathered responsibly and analyzed to the best benefits of the trial. Of course, you can't train on everything, and I think I will leave this slide just for review after this webinar, as we're running a little bit out of time. But there are guidelines by the FDA issued in January, European guidelines, which really helps us to understand how to best deploy AI. And imaging call-ups, imaging CROs probably can provide enough expertise to guide you into what to do and what not to do when it comes to planning a trial, which will include AI. Last but not least, I think we thought about what CMOs and operational leaders can do now. Firstly, of course, you can just pilot AI-assisted analysis as an exploratory endpoint in the next trial. There is no need to use AI as a primary endpoint without full confidence. You can discuss with us how to build DICOM standard data pipeline, which will last from very first phase 1 all the way forward. Fitting that retrospectively is not cost beneficial. You can also look at the endpoints which you deploy in the next trial and think beyond of some of the points which were used before or done before, but really think what could make sense for your own mechanism of action and your own trial. We definitely can help you plan your data governance and help you to put it as a part of the protocol. And lastly, of course, is choosing an imaging partner who can help you to scale from phase 1 all the way to phase 4 and make it compliantly and consistently with the best use of technologies. I really think that, the next biotech is not going to be the one who has the most imaging data. It's going to be the one who is really using the right models, the right tools, and just deploying them on the imaging data that you collect in your trials. On that, I will stop and invite you to contact our team. So you have our email, you have our website. We, of course, would be delighted to give you more examples, show you how we work in trials, how the AI could be complementary to the trials that you run. And I know we have a few questions. If we have time, we can answer maybe 1 or 2. I'll just open the chat. And otherwise, we truly thank you for your time today, and hopefully you will reach out with all of the questions. Oh yes, I see quite a few questions. So I will start from the top and do that for a couple of minutes. So we overrun by 5 minutes, and then we close at 10:02. So the first question is IAG specifically focused on trials or involved in solutions which are not related to trials, so complementary solutions providers? I think it's a great question. So we work with endpoint providers who do very, very specific and very targeted endpoints. DYNAMIKA allows us to incorporate those new methodologies, new algorithms through API or through a slightly different pipeline. So from a sponsor point of view, there will be an opportunity to really have a single point of data management, but still deploy all of the novel techniques which are available. I will move to the next question just for 1 minute, and it is from Yazdan. Thank you for the very interesting talk. Thank you for listening. What is your insight on foundational models that combine all modality imaging, histopathology, CT, PET information? Those are extremely interesting. The foundational models, if you guys don't know, it's a way of bringing different imaging modalities to answer a specific question. I think in trials, it might be a little bit of a challenge because the models might not be designed for a small data set, which we normally encounter in early phase trials. So perhaps when we're a little bit more mature with data strategies and we have more data to deploy, we can certainly start looking at more foundational models, overarching models to get some insights. And I think those are brilliant questions. So I will stop here. Thank you all for joining, and we hope to see you again. We're going to run another webinar shortly. Thank you.